ever wonder what's going on, like science-wise, inside a rapid antigen test? Well, you know I did, so let's take a look.
So here's the gist for those who want it, and then I will get into the details. So the basic idea is that these antigen tests are going to look for a viral protein, typically the N protein, which is this little blue guy that it uses to cover the RNA. It's not really blue, it's just colored blue. But anyway, so the basic idea is that these tests are going to look for it, typically in a sample from your nose. They're going to break open the cells and break open the viruses within the cells. And the basic idea is that they want to look and see if that protein's present. And how they're going to do that is using labeled antibodies. And so we'll get more into this. The antibodies are just these little proteins. And so our body can make them against various things, but we can also make antibodies in the lab and we can attach them to labeled things. And this will allow us to capture antigens. So an antigen is just something that an antibody binds to. So the cool thing about antibodies is that they can specifically bind to things. So we can get antibodies that will specifically bind to the N protein, and then we can conjugate or attach those antibodies to like a colored bead. And this way, when you apply the sample from your the broken up open cells and broken open virus from your nose sample, and you mix it with these labeled antibodies, if the N antibody is present, then the antibodies are going to stick to the N protein. And then if you have another antibody on your strip, and so you have an antibody that binds specifically to the N protein, you can get this like antibody sandwich where you have the antibody bound to the N protein at like two different sites. And so this will allow you to get your beads stuck onto the pad, and then you'll get a band. There's also like a control antibody, a different type of antibody that'll get stuck at the second line. And so you should always see a line at your control line. And this is all going to get whipped through the pad thanks to a wicking pad at the other end. And so there's basically, you apply your sample on one end. So you have this like sample pad where you apply your sample. It'll look like this is hidden in behind the hole. Then what's going to happen is you have a line, your test line is going to be somewhere, and then your control line. And so on this test line, you're going to have the anti-N antibodies, so against the N protein. And then in your control line, you're going to have antibodies against the control, which can be, I think, so sometimes they have like a chicken antibody or something, and then an anti-chicken antibody, but it's just some like colored antibody. And so what's going to happen is then on the other end, you have this wicking pad. And so this is going to help through like capillary action, basically the idea that water is going to stick to each other. It's going to stick to these things like it's going to be kind of like pulled through, kind of how if you like stick a paper towel in water, it's going to wick the water through. And when it's wicking this liquid through, it's going to be pulling through the things that are in it. And so it's going to be pulling through those beads. And if they're labeled to N, they're going to get stuck on this anti-N line. If they're not, they're just going to flow through into this wicking pad, and they're just going to end up down here. But if there is N protein, then it's going to get stuck down here. And so you'll see a positive test. You'll get a test line on your test line, the T line. So a little more detail. So basically, SARS-CoV-2, the virus that causes COVID-19, it's a single-stranded RNA virus. So it has one strand of RNA and it's coded in this nucleocapsid protein N. And so this is the one that these tests are looking for. Surrounded by a membrane envelope, and it has these envelope proteins. And the protein that you're probably most familiar with is the spike protein. And so the spike protein is going to jet off from its surface, and it's going to allow the virus to dock onto cells through this ACE2 receptor. Then either at the membrane, or it'll it'll either merge at the membrane or it'll come into the cell in like this membrane pouch called an endosome. And then the spike protein is going to undergo this like shape shift that's going to help it merge with the cell membrane and dump out the viral contents so that our cells will start making more and more of the virus. And so basically, when you're infected, you're going to have this virus in your cells, and your cells are going to be making a bunch of this virus, and your cells are going to be shipping out a bunch of this virus. So you're going to have a bunch of this virus, and so you can look for parts of the virus to see if a person's infected. And so the tests that like the typical like gold standard tests or whatever, that are like these PCR tests. And so basically those are going to look for the viral RNA, or at least they're going to make DNA copies of the RNA and look for those. So typically how these work is you extract the RNA and then you make lots and lots of copies of the viral RNA that's there. And you have this in a way that you're going to be able to detect the copies as they're made. And so if you don't have any of the viral RNA, you're not going to make any of these copies, and so you're not going to get a signal. But if you do have the viral RNA, then you're going to get this a signal. If it goes across the threshold of the noise, then you have to say it's a positive test. So this is looking for the viral RNA. But that's not the only thing you can look for. And so these antigen tests are looking for the viral proteins. And so typically they're looking for the N protein. Because it's really, really abundant, and it is going to be less sensitive and stuff to mutations in spike, and it's easier to make in the lab, and so to do all these tests and work with and stuff. The mutations in the spike protein aren't going to affect these tests, although there could potentially be mutations in the M protein or mutations that cause the virus to make more or less amounts of different proteins that could affect how these tests work. But the mutations to spike itself should not play a role. And this is why we don't need to worry about these tests not being affected by mutations to the spike protein. The terminology can get confusing because we use an antigen test that uses antibodies but looks for antigens. So you might also hear of like serological tests or antibody tests that are looking to see if you have antibodies using antigen. And so let's go over these terms. So basically antibodies are just these little proteins that can be made by the body in this sort of like trial and error approach, where basically your immune cells, these type of immune cells, these special B cells, they're gonna make, they're each gonna like choose randomly a different antibody to make. And so they have these different like variable regions. And so the ones that choose something that actually binds, they're going to get selected for. So if an antibody just happens to have a combination of variable region segments so that the protein has this has the properties that it's going to bind to some foreign thing, then it will bind and it'll get selected for. So the thing that it binds is called an antigen. And so we have the antibody is this like protein, and so it has this constant part in these generic parts. This constant like generic part, and then these variable parts. And so these variable parts are what's going to be binding to the antigen, and these are going to be the parts that the cells kind of experiment with to find ones that bind. And so anything can be an antigen, but typically you want your own body to just make antigens against foreign things, not your own self things, or else you can get like autoimmune diseases. You can also get antigens against things like peanuts, and so people can get allergies, various different things. But the basic idea is that the antigen is the thing that the antibody binds. So if you want to see if someone's like had the virus, or maybe if they have like response to a vaccine or that sort of thing, you can talk about like antibody tests. And so these are going to test to see if your body has made antibodies against the viral antigens, so against the viral protein. When we're talking about an antigen test, we're looking in like reverse. So we're using antibodies that we make in the lab in order to test and see if the viral antigen is there. So if the viral protein is there. So we're using labeled antibodies to look for antigen. The various labels can vary. Sometimes they're like colored beads, so like latex colored beads or fluorescent beads. Sometimes they're like colloidal gold. And so basically, if you have like, it's not like, if you have like really, really, really tiny little gold like in a solution, what's going to happen is the gold has a lot of electrons, and it goes this cool thing called like surface plasmon resonance type of thing, where basically the light waves can interact with the electrons in the gold, and long story short, it gives you this red color. And so this is often used. But the basic idea is that somehow your antibody is going to be labeled. And sometimes they're like fluorescently labeled, but then you need a special detector. But because it's labeled, you're going to be able to see where it ends up. So you have the antibody that's specific for N, and then you also usually have some sort of like control antibody. And this control antibody is going to help you know that the test like is functional, so you know that everything got worked through, and that the antibodies, at least these antibodies, were working. And so this is why you should get 2 lines, and if you get like just a single T line but not the control line, then there's something wonky with your test. So the test starts, basically you have to like swab your nose, and then you stick it in this like buffer. And I'm not like advertising for this test or anything. This was just like literally the only test that I could get. And it was a pain to try to get it. But anyway, I'm not going to go into those problems right now. We need more of these tests, basically. But the idea is that when you're basically you have this, it comes with this tube with like this buffer, this lysis buffer. And so lysis is when we like break open cells, or like when we break open viruses. And so basically it works kind of like this lysis buffer is going to work kind of like soap. And so it's going to disrupt the viral membrane. It's going to disrupt your cell membranes. Basically, it's going to just, all those are surrounded by these lipid membranes, these like oily membranes. And so you can disrupt that membrane, then you can break things open. I'm not exactly sure what's in these buffers, but the idea with soap is that you have some sort of like detergent or soap that's going to break, like kind of slither into the membrane and kind of disrupt it and get things to break open. And you need those things to get broken open because remember the N protein is going to be like inside of the viral membrane. It's not going to be like the spike protein that's on the surface. So when this and then so you need, then when you do that, you apply your sample to here. And once it's on that conjugate pad, so basically these are going to have like these antibodies, they're going to be like dried on there, but they're not going to be like permanently stuck. Like these guys are like permanently stuck. These guys are just kind of like stuck because they're dry. And so when you put the liquid on there, then they're going to dissolve and they're going to mix with whatever's in the sample. Once they're dissolved and there's in this liquid, now they're going to flow through to the wicking pad, and then you look and see where the labels are caught. So it's going to take longer for a person to start to be positive by an antigen test than they would be with like a PCR test. And people will probably stop being positive on an antigen test before they stop being positive on the PCR test. And so but this like lack of sensitivity often isn't as bad as you might think, because it's most sensitive at telling if people are like probably infectious. So basically, if you're making enough of the virus so that we can detect the protein, then it's likely you're infectious. Whereas if you are like far out from getting the virus, you might still be shedding just some like dead virus and pieces of viral RNA. The PCR tests are really, really sensitive, so they might be able to pick that up. And so this is why people can like be testing positive for a really, really long time based on those PCR tests. And so they're not typically used to tell if a person is still contagious, whereas an antigen test is a better idea of if a person's contagious. It's not like totally 100%, but it gives you a better idea. And because these tests are easier and cheaper, then if people can do them more often, it's a better like public health strategy on a lot of fronts. Problem is that it's really, really hard to get these tests. And yes, they do, the lower sensitivity can miss infectious people, but it is definitely better than nothing. And it's definitely better than waiting like 6 days to get your PCR test